Hello, everyone, and welcome to the American Brain Foundation's The Next Generation of Brain Research. We are going to take just a few more seconds because we are still getting people in. So grab a quick drink, get yourselves settled, and we will be with you very shortly. Okay, great. Hello, and welcome to the American Brain Foundation's annual Meet the Researchers event. My name is Kelly Prezen, and I work with the American Brain Foundation. For those of you who are unfamiliar with us and with our work, the American Brain Foundation funds research across the entire spectrum of brain diseases and disorders, including Alzheimer's and other dementias, Parkinson's, MS, stroke, epilepsy, cognitive aging, peripheral neuropathy, and more. We know that all brain diseases are interconnected, so funding across the full spectrum of neurological disease is our best hope for finding improved treatments, preventions, and cures. Brain disease is a global issue, with 1 in 3 people around the world affected by it, whether they are a patient with neurodegenerative disease, a caretaker for a loved one, or a researcher working on one of the hundreds of diseases that have a staggering effect on our lives. We at the American Brain Foundation, and I trust many of you, have heard a great deal about cuts to the National Institute of Health's research funding. According to a July 2025 article in Science, the NIH is already $6 billion behind in spending money that has been allocated to it for research. And there may be more potential cuts on the way. So that's what makes grants like the Next Generation Research Grants and our Cure One, Cure Many initiative so significant and important. As I'm sure you'll hear tonight, they help researchers launch a career and investigate new and novel approaches to diseases. I hope that you will consider supporting these researchers and future researchers by making a gift to the American Brain Foundation at any point during or after this presentation. We're going to make it easy for you. We have a QR code. There it is, right there. So if you are on a laptop or a desktop, you can take a quick scan of that and head on over to the American Brain Foundation, make a donation. If you are watching this on a cell phone, you can find us at americanbrainfoundation.org. And the donate button is right there for you to find. So I would also be remiss if I did not thank our program partners who collaboratively fund these grants with us. We truly would not be able to make these grants without them. So a huge thank you to our partners who you see on the screen in front of you. Also, thank you to all of the individual donors who make these grants possible. Okay, on to what we're really here for tonight. I have spent the past few weeks interviewing these researchers in order to get to know them and gain a layperson's understanding of their projects. Every single one of them is an up-and-coming expert in their field, and I'm not the one who's decided that. That's been decided by scientists. So they are, in addition to being these experts, they are universally engaging, passionate, and thrilled by the idea of being here to tell you more about their research. They exemplify the Next Generation Research Grant Program. So the Next Generation Research Grant Program funds and supports innovative investigations by the best and brightest early career clinician scientists, including the ones you'll hear from shortly. Over the years, this program has provided over $45 million to over 300 researchers. Those researchers have gone on to have their career scientific findings cited almost 1 million times. I just want to say that again because it's so mind-blowing. So we have funded over 300 researchers whose clinical career scientific findings have gone on to be cited almost 1 million times. So tonight, each grantee will provide a short overview of their program, and then I will ask them some questions to get to the heart of why they decided to go into this kind of research. It builds incrementally, and it's not always the most lucrative or glamorous career to be a research scientist. We will talk to them about their passion and their drive, how they handle failure, what gives them hope for the future of research. And after our guided conversation, we'll have time for a Q&A from the audience. So please submit the questions you have in the box on your Zoom screen. Okay, I am so excited to introduce you to our researchers. And let's begin with Laura, whose research involves neuroimaging involuntary movements known as dystonia and Parkinson's disease.
Hello, can you hear me? I'm stuck in an airport in the restaurant, so there might be some chatter, but I really wanted to make it here. Thank you so much. And so my research is on dystonia and Parkinson's disease. So Parkinson's disease is the second most common neurodegenerative disease, and it affects a huge amount of people as our population is aging more and more. And these people, they have a lot of different symptoms like tremor and slowness. And dystonia is these involuntary posturing movements, twisting that can be really painful and debilitating. And we don't have a lot of good treatment for this, even though it affects a third or even more of the people with Parkinson's. So I'm an expert in neuroimaging, and my project is to use neuroimaging to understand which brain networks are affected by dystonia in Parkinson's so that we can better develop treatments for dystonia in this population.
That was great. Laura, can you tell us what institution you're at?
Oh, yeah. So I'm a movement disorder fellow, which is I'm a neurologist studying to become a movement disorder specialist and treat Parkinson's at Washington University in St. Louis. I'm on my second year.
Excellent.
Really appreciate the tip.
Perfect. Okay, I would like to move on to Peter, who is investigating epilepsy, and I think I've got this right, developing a brain atlas for neural stimulation. Did I get it, Peter?
Yeah, roughly. Thank you, Kelly. And thank you to the American Brain Foundation for all their support. So my basic idea is that I want to try to improve neurostimulation for epilepsy. So just to give a brief overview, seizures are these episodes of abnormal electrical activity that can sometimes affect thinking, sometimes have external manifestations like convulsions. Epilepsy is the disorder that's associated with seizures. And most patients are well controlled on just medications, but about 1/3 require brain surgery. And then some of these can't get the resective brain surgery, so the cutting out of the brain. And these patients will need neurostimulation to achieve some amount of seizure control. However, it's still not perfect. And one of the reasons that we worry that it's not perfect is that we haven't translated all our findings from research into clinical practice. Specifically, in the past, we used to think that epilepsy was just this lesional problem, that a single part of the brain is abnormal. And if we just remove it, basically that's a cure. It turns out that that's not the case, and the brain is much more complicated. And it turns out that there are networks in the brain, and you actually need to basically attack different ones to try to control seizures well. The problem is that our neurostimulation approaches clinically still focus on this lesional approach where we just hit a target as opposed to looking at the whole brain holistically. So my idea and my thought was that we would actually use these virtual brain models based on neuroimaging, based on brain waves from EEGs, based on neurostimulation testing data that we can then individualize and personalize patients' epilepsy neurostimulation care. I'm currently looking at about 150 patients with epilepsy who have gone neurostimulation, and I'm trying to look at structural imaging, functional imaging, intracranial encephalography, and some of this neurostimulation testing to try to better understand their epilepsy. And I really hope that with the work that we do, we'll be able to find and develop therapeutic neurostimulation that is more methodological and is more that can be curative for our patients.
Awesome. Peter, that was perfect. And can you tell everybody what institution you're at?
Yeah, I'm currently a physician. I'm an instructor at Mass General Hospital in Boston.
Fantastic. Okay, so Tiffany, I have you up next. And Tiffany is joining us all of the way from Sydney to talk about peripheral neuropathy and imaging. So good morning, Tiffany. We're delighted that you were able to join us today.
Yes, good morning. Yeah, I know peripheral neuropathy is a bit of a mouthful. I think I've had to get used to it over the years. But yes, I'm Tiffany from the University of Sydney in Australia. So thank you so much for having me at a reasonable time zone today. And my area of interest is in chemotherapy-induced peripheral neuropathy. So just as a bit of background information, in recent years, cancer treatments has advanced such that we have more and more cancer survivors living for longer, which is really good. However, a lot of the chemotherapies that they've been treated with can actually cause permanent nerve damage in the hands and in the feet. This means that even after they've completed all their cancer treatment, a lot of patients are left with residual sensations in the fingertips and the feet, and that gives them quite a lot of disability, including things like manually fine motor tasks, turning keys, picking things up the floor. We've had patients that aren't able to drive because they can't feel the pedal in the car in their feet. And all this is really debilitating. Unfortunately, it's chronic in a lot of these cases, so these symptoms persist with them for the rest of their lives. So even decades post-treatment, they're still left with these symptoms. And unfortunately, at the moment, there are no treatment or preventions for peripheral neuropathy beyond reducing the treatment dose as the symptoms arise during treatment. And while this isn't ideal, it's the only thing that we can do. However, even then, this really depends on our ability to pick up these symptoms really early on so that we can implement this intervention as soon as we can to prevent this development of chronic and severe nerve damage. However, at the moment, we just really don't have the tools that are sensitive enough to pick up these changes early on. So my research study is looking at identifying new biomarkers that are able to identify the earliest changes in nerve damage and to be able to catch these symptoms early on and help patients in preventing further damage down the course. So I'll be looking at two different types of biomarkers. One of them is a blood-based biomarkers. So there are proteins that are released into your blood when your nerves get damaged, and we want to see how sensitive these measures are and picking up symptoms as soon as possible. And the other one is a dermal imaging. So we're using a new technique to image the nerve endings at the fingertip to be able to quantify how well your nerves are going. And hopefully these two can be implemented as early biomarkers of nerve damage.
Excellent. Thank you, Tiffany. I have a very beloved friend who was an excellent embroidery. She did beautiful embroidery, and peripheral neuropathy has affected her ability to do that. So I have a real personal connection to your project. So, okay, moving on, we are going to talk to Giovanna, who is taking a public health approach to cognitive aging and age-related memory loss.
Okay, I need to mute myself. Well, my research is about something that is important for everyone. Preserving brain health is crucial at any age. Lifestyle factors like exercise, diet, and smoking can impact brain health as we age. In addition to our lifestyle, depression and anxiety can also cause memory problems, trouble with thinking, learning, judgment, and increase a risk of Alzheimer's disease. It's important also to consider that previous studies have found differences in diet, alcohol consumption, smoking, physical activity, and the frequency in reporting depression and anxiety between women and men. However, these studies have explored lifestyle, mental, and brain health separately. What I'm doing with my study, with my research, is studying how this behavior influences memory and thinking along with their effects on mental health. I will also examine whether men and women experience these effects differently. What is important, because I think that findings from this research will help us to design and implement interventions and programs to maintain brain health, prevent or slow memory and thinking changes in older adults who might be at greater risk, and also to consider the impact of these factors in the success of the programs that we implement.
Thank you, Giovanna. And let's see. Now we are going to move on to Deborah, who is exploring adverse childhood experiences, also called ACEs for anybody who has ever done any youth development or psychology work, and their impact on age-related cognitive decline.
Yes, thank you very much. It's such a pleasure to be here. Good evening, everyone. So the work that I'm doing, and I'll actually briefly introduce myself. So I'm a behavioral neurologist at Johns Hopkins, and much of my clinical work is seeing patients with memory disorders and other cognitive impairment concerns. And so my research is more epidemiologic. I'm focused on understanding the relationships between early life adversity, so traumatic exposures that occurred before the age of 18, and how that impacts cognitive performance later on in life. Much of my work has also looked at other measures of cognitive outcomes as a big bucket. So neuroimaging measures of white matter disease. So looking at how certain vessels in the brain have developed plaque over time, and is that influenced by extreme stress and significant trauma early on in life. And we also look at biomarkers of Alzheimer's disease and other neurodegenerative disorders. So another big piece of this is trying to understand what may explain that relationship between what we're seeing with early life adversity impacting cognition in older adulthood. And so we are focusing primarily on measures of inflammation primarily in the blood. But we also look at some neuroinflammatory measures as well to see if that helps to explain that relationship between early life adversity and poor cognitive outcomes. The hope in the long term is to actually develop, this is a long-term goal, but to really develop a screening tool that we can implement in memory clinics and primary care clinics for middle-aged and older adults to identify specific factors of stressors that occurred early on in life, as well as in middle age, and some of the sequela of those stressors, including depression, loneliness, things that we can actually intervene upon to help prevent the risk of dementia.
Okay, thank you, Deborah. Now we have, I keep clicking away from my script where I have everybody's program summarized, so you'll have to excuse me. So now we will have Arends tell us about his project involving ALS and the brain's ability to regenerate.
Thanks, Kelly, and thanks for this invitation. So I'm a newly appointed faculty at Johns Hopkins. Also thanks to the support from the Brain Foundation and the AAN. My clinical and research interest is in ALS, or Lou Gehrig disease, or motor neuron disease, fatal neurodegenerative disease that affects limb muscles and the respiratory muscles, that is invariably fatal. The reason for that is because we fundamentally don't know the mechanism of the disease, and my work tries to tackle this question. Specifically, I focus on a protein called importin beta-1, which I and others have identified to be involved in restoring nerve function and promoting neuroregeneration under normal conditions. In this project, I try to, and I hypothesize, I try to verify if this protein is actually impaired in ALS, due to clumping inside the neural cells, and whether this impairment leads to decreased ability of the nerve to regenerate after damage. One central goal of the project is to see if by promoting or enhancing the expression of this protein in the neurons of ALS patients, we can promote the ability of nerves to recover and in that way promote motor function. I tried to do that using cell-based models of the disease, which are quite advanced and being developed in the last decade or so, and kind of chips that recreate the structure and the organization of the motor pathway in vitro.
Okay, thank you very much, Arends. Okay, last but not least, we have Joe. Joe, my notes on your interview are a little bit scattered, and I have the words zombie proteins written down, and how those affect folks with ALS. So, am I getting that right? Are you studying zombie proteins?
In a way, Kelly, in a way. Yeah, so hello everyone. I'm Joe. I'm at the University of Missouri in Columbia, Missouri. I'd like to thank the American Academy of Neurology, American Brain Foundation, and ALS Association for this great honor. Well, as we age, all of us begin experiencing a gradual loss of strength. Our muscles weaken, daily activities become harder. So a central focus of my research is to understand why this happens. But I also study diseases that cause extreme accelerated weakness well before normal aging. Diseases like amyotrophic lateral sclerosis, or ALS, also known as Lou Gehrig's disease. So ALS is a motor neuron disease, meaning that it selectively degenerates the neurons that allow our muscles to work. In ALS, muscle wasting progresses very, very rapidly, leading to complete paralysis and tragically death within 3 to 5 years after diagnosis. Today, there are no cures and only very limited treatments. So what makes ALS especially striking is that beyond this obvious muscle loss, we're finding signs of accelerated aging, but accelerated aging at the cellular level. So cells in the nervous system appear older than they should be in ALS patients and in ALS mouse models, which may help drive the devastating decline we see in patients. So with the support of this grant, I'm currently investigating whether we can improve aspects of this cellular aging. So if we can restore youth-like function to vulnerable cells in the nervous system, we may be able to slow or even improve symptoms in ALS. So with the support of my mentor, Dr. David Arnold, and my fellow lab mates, we already have exciting data that suggests that this approach might be possible.
All right, Joe, thank you. If you are like me and you listened to all of these amazing summaries and have a million follow-up questions, I would really encourage you to put them in the Q&A because we will get to them in just a few minutes. But right now, we're going to start with a little bit of a directed conversation. And I am going to start with, okay, this is a question for Peter, Deborah, Joe, and Tiffany. And you can unmute yourselves when you are ready. Or if everybody is a little bit shy, I will end up calling on one of you. But my question is, can you tell us how your next generation research grant impacted your career trajectory?
I'm happy to go. So I would say first that this opportunity has allowed me to take on a project that allowed me to really interrogate those biological mechanisms. So really digging into the biology of an individual who may have experienced significant stress and trauma, and see how that can impact their cognitive performance in middle age and later on in life, in older adulthood. And so, because these have been really interesting questions we've been asking, and finding actually some interesting things that hint more toward resilience, so actually refute our hypothesis that inflammation, and at least in the cohorts that we've looked in, we hypothesized that inflammation would drive down cognitive performance, but in a couple of large data sets of thousands of individuals, one of which was of older Black adults in Chicago, we actually saw in this particular cohort that those folks who had higher levels of inflammation did better on certain cognitive domains. So what we found spurred on a lot of additional questions. And actually, this is what the work that I've been doing, thanks to this grant, has propelled me forward to ask more of an intervention type of question, to looking at developing a screening tool that can get more at some of these resilience factors that we can play upon and sort of target for people to help them reduce their risk for dementia in older adulthood.
That's amazing, Deborah. Thank you for sharing. Peter, I see you are unmuted and ready to go.
Sure. Yeah, I'd say the support has been essential, especially in this this day and age where funding sources, especially from the government, can be a little uncertain. ABF and AN and AS, in my case, their support has really helped propel my research forward. And my research is translational as well, which can be a bit more difficult to get funded through traditional sources. So it's actually been very useful for me to bring together my expertise in imaging and neurophysiology to try to improve clinical care. And some of the support has allowed me to ask additional questions on stimulation and to better understand how stimulation affects the human brain. So both from a translational standpoint and also from a basic science standpoint, ABF support has been amazing.
Yeah, Peter, could you do me a quick favor? Can you tell me and our audience what do you mean by translational research?
Sure. So in broad scopes, you can think of basic science research as the scientists working away in a lab doing cell cultures, for instance. You can think of clinical research as maybe a physician going through charts and trying to see links between different diseases that are kind of more maybe from a statistical point of view. Translational research tries to meld the two and bring things from the term they like to use is from bench to bedside. So you're taking stuff from the lab to the patient's bedside, and you can translate research directly into clinical care.
Fantastic. Thank you. Okay, I had Tiffany unmuted first. Joe, she beat you, so we'll do Tiffany and then we'll do you.
Lovely, thank you. Yeah, I echo Peter and Deborah's thoughts exactly on how great this opportunity is. I think, especially as an early career researcher, I'm only a few years post my PhD. There are really limited opportunities, and grants like these really allow us to establish ourselves as independent researchers, and really branching away from our professors, our supervisors, and our teams a little bit to be able to take on our own project. It's very validating on a personal level to be able to do that, to follow the passion and the questions that we have. I think this intellectual curiosity is what drives a lot of research careers. So being able to obtain the funding and get the grants to be able to do that at this early career stage is really crucial. And I think it's really important for us as early career researchers to have these opportunities. Because I think this is the time where a lot of people might give up on this kind of career because it is really hard to get funding. But I'm very grateful for our funders and giving us this opportunity to take this big step. And hopefully we're able to then take that and move forward and on to the rest of our careers with this.
Perfect. Thank you, Tiffany. And for those of you who are not familiar with the American Brain Foundation, we do fund globally. So Tiffany is down in Sydney and is one of our researchers. So not just in the United States. So if you are a potential future researcher watching this webinar to glean information, know that we will fund globally. And with that, Joe.
Yeah, I'd like to second what Deborah, Tiffany, and Peter just said, that this support has been just fundamental and important. It's really been like a seed almost that has allowed not only me, but other folks in my lab to generate a lot of other projects that have stemmed from the initial project that was funded by this grant. That can hopefully help us understand ALS a little better and hopefully come up with treatments. So my main focus, for example, is when I joined my current lab, is how aging affects the nervous system and to better understand why it is that as we age, we get weaker. But as you know, this grant has opened even more doors and opportunities to investigate, this muscular decline, but caused in different contexts. In this case, the context of neurodegeneration like ALS. And so one striking outcome of being able to study both weakness in the context of normal aging and neuromuscular decline in ALS is that we're beginning to understand that there are many similarities between uh, age-related weakness and many aspects of ALS, and even potentially shared mechanisms, which is honestly quite intriguing, is what we're starting to find. And so, why is it significant? Because understanding one might help you understand the other. And to sum it up, for my scientific career, this grant has given me the most wonderful opportunity to connect dots, so to speak, across these different contexts like aging and neurodegeneration, with the potential of honestly improving both.
That's really wonderful to hear, Joe. As I said before, maybe I didn't say it before, one of the American Brain Foundation's goals is to cure one. We believe that when we cure one disease, we will cure many. And we know that the work that each of you are doing in your specific area will have ripple effects out into other brain diseases. So, okay, with my next question, I would like to talk to. Oh, okay. So for those of us who live and work sort of in, I don't want to say mundane work, but we're office workers, we are not scientists. We have maybe a Marvel comic Spider-Man view of what a researcher does. So I would like to hear from Arens and who's my other person who got very excited about this? Laura, to tell me about a day in the life of a clinician researcher. What does a typical day look like for you?
Yeah, a lot of accidents. So I mean, the kind of work that I do is that will be defined of a clinician scientist. So as Peter was mentioning, I go from seeing clinic, for example, this morning I had my clinic patients, right? And then in the afternoon, I had to run to go to the lab because I had to feed my cells, which is like to actually give them water practically, but a very special kind of water. And so it's a lot of multitasking, and where the clinical part has a very significant burden. It's actually the majority of the burden that we have in terms of time. And this grant are very helpful to kind of protect a little that space that we try to keep preserved for the lab work, right? And so a lot of multitasking. When we go to the lab, there's a lot of failures. Frequently things don't go well. I mean, it's kind of the vast majority, they don't go well. Sometimes they go well. Sometimes they go against where our hypotheses were, which is great. We change our mind. We are very flexible. We question ourselves frequently. So a life of uncertainties and running, a lot of walking.
Yeah, absolutely. I like to, second what Aaron said. And I think also to me what's exciting and challenging is knowing how to wear these different hats. So when I'm wearing my clinician hat, I have to be very confident and I have to tell patients and their loved ones like what I think, is happening and what I think are the next steps. And then when I'm back in the lab, I have to be very uncertain and very questioning because we want to get the best data possible. And so this change in pace, and then also the change in pace of like this very slow development of science that one day might help someone, you hope many people, but many times, you go down the wrong track for a couple of years before you find the right track. And then back to the clinic where, you help a lot of people in one morning. This morning I was just doing injections for patients with dystonia and, in the morning I saw 20 patients and helped 20 patients. So it's keeps you motivated. One side keeps you going on the other side.
Okay, awesome. I'm sorry, I'm doing a webinar and I am trying to rein in my enthusiasm about all of your projects and asking questions that have occurred to me since the last time we talked. So sticking to what I have actually written down here. So I'm curious about how folks got involved in their specific field of study. And Giovanna, if I'm remembering right, you actually came to us via dental school. So I'd like to start with you.
Yes, that's true. I'm a dentist trained in Colombia, but I came here to pursue a master's in public health and epidemiology. And as a graduate student, I started to work in the department of neurology. And by collaborating with all these researchers about non-pharmacological intervention, about Alzheimer's disease and related dementia, risk assessment is like, okay, this is really interesting. And that kind of really guide me about new research opportunities, about getting more knowledge about data management approaches, and understanding how this research that we do, we can translate later from clinical settings to community-based settings, and then we can impact more people and help more people.
Okay, excellent. Joe, can you tell us how you ended up in your specific field of study?
Yeah, absolutely. My main research interests that I currently have now actually started with crabs. So kind of weird. So during my PhD, I actually investigated a very small network of neurons in crab stomachs. So this is a small network made only of 26 neurons. And so in these animals with these very simple nervous systems, I was able to ask very basic and fundamental questions about how neurons work. So for example, how can neurons do their job for a long time? And why do neurons fail at doing their jobs sometimes? And so during my current postdoctoral training with Dr. David Arnold, I've really kept asking the same questions I was asking crabs, but in the context of human disease. And in this case, ALS, and apply to mammals, to mice specifically, to answer similar questions in the context of ALS. So why do certain neuron types in ALS fail, and how can we improve the function of those networks, those neurons in ALS?
Started talking before I unmuted. It's amazing to think that your research started with crabs and is now being translated into mammals and hopefully into people. It's wild to think that that's where some of our science starts. Okay, so I'm going to move to Tiffany. Tiffany, you and I talked at length about how these NIH cuts have been affecting your work because we would think that as you are not in the United States, it wouldn't have a dramatic impact on you. But you corrected me on that. So I'd love to hear about how funding cuts in the United States are impacting your work.
Yeah, absolutely. I think in this day and age, there is so much international collaboration on various different levels, even us on the other side of the world in the Southern Hemisphere, are definitely impacted as well. I guess for one, as I said, we have so many colleagues around the globe, and we're always concerned about how they're doing, how their funding situation is. We know that if they do get affected, that is definitely going to have a downstream effect on how much we can collaborate. The projects that we've already set up, are they going to still be able to continue? How that would change things. But also practically speaking, a lot of the grants that go around do come from the US. And really, when you minimize the pool of funding that's available, naturally that means less opportunities for us as well. I think a lot of US funders are going to prioritize, and rightfully so, US academics because of the limited funding that's happening. And that means less to go around for other parts of the world. And just for the different grants that we apply for, there is obviously reduced supply means increased competition and harder to get things. I think overall, just less investment in research in one country is really going to impact globally or everyone that's going to be looking for funding. But also, I think, especially in this early career stage, I think limited funding would just mean that there's almost a generation of early career researchers that miss this opportunity to embark on this career that they might have considered. And overall, I think science is going to perhaps suffer a little bit just with less minds having the opportunity to get started in the field.
All right. So one of the things about the American Brain Foundation is that we can only fund 1 out of every 4 grants that have scientific merit. So that means for all of the researchers that you see here, all of the researchers we fund in a year, there were 75% more researchers who had promising research that we just couldn't fund because we didn't have the money. So we would like to ask again that you consider scanning our QR code and donating some money to the American Brain Foundation and help us to increase the number of new researchers on this call by 75% so you can hear about how their lives started in dental school or working with crabs, or you can hear them talk about the best parts of their jobs and what gives them hope. So that's actually my next question, and it's for Deborah and Laura. And can you tell us what the best part of your job is?
Sure. Yeah, I'm happy to share that. Actually, I would say that a lot of what I feel is the best part kind of piggybacks off of what Laura already shared. Because I'm also a physician scientist, and I feel that much of the work that I'm doing in the clinical space is really connected with what I'm doing in the research world, which is rooted in epidemiology. So I'm looking at large data sets of many, many people and looking for patterns that are not very obvious on the surface. So doing a lot of advanced statistical modeling to really answer questions that come up all the time in my clinical encounters with patients. And so honestly, I feel that one of the best parts of what I'm doing is bringing some of those findings from what I'm reading or what I'm from my own results and my work to my conversations with patients, especially those who are really struggling with current psychosocial stressors and are at very increased risk for cognitive impairment or dementia because of those stressors. And so I find it really rewarding, and I'm very excited about the next steps that our team has outlined to really rigorously intervene for these patients.
Yeah, absolutely. I think, a lot of us here see patients with chronic conditions that don't have cures. And so, one of the good sides, we develop a relationship. We see these people throughout many years, and it's lovely to learn about who they are and what motivates them. It's also very sad that we don't have something that can make them better, that can cure them. And then being able to, tell your patients about the research that's happening. And so many times, people are just wonderful and they want to participate and they want to help, and you can recruit. And to be able to give that hope and, to continue to hear their voices so that you can really shift science in the direction that they want it, that they need it. I think that's the best part of being a clinician scientist.
Fantastic. And I just want everybody listening to know that everybody had a great answer to that question. And I wish we had time to let everybody talk about their passion for their work because it's really just an amazing thing to be able to see. So, uh, instead, we are going to continue on, and I have a question for Peter, Joe, and Giovanna, and it's what gives you hope for brain disease research?
I think the thing that gives me the most hope are probably the people that I work with, both at my institution at MGH and the larger American Epilepsy Society and community. I have a chance, and as a clinician scientist, as many people on the call, I have an opportunity to see some of what I'm working on, which is responsive neurostimulation in clinic, every other week, basically. And I get to see patients actually benefit from some of the stuff we're doing. And I'm surrounded by incredibly intelligent, hardworking, passionate clinician scientists and scientists as well, who are all devoted to not just helping patients, but also understanding the brain. And, some of the stuff we discover, even though it may not help patients now, it may help patients, 20, 50 years from now. So I'm always kind of in awe of the people around me. And I'm incredibly fortunate to just be in this environment, in this community, and have the support of the American Brain Foundation.
Yeah, go for it, Joe. I saw you unmute yourself first.
Yeah, so one of the very eye-opening and rewarding aspects of the research that I do now in ALS is that I had the fortune, the privilege of meeting folks that are either affected by neurodegenerative diseases like ALS or have family members that are affected by it. And when we've gone to fundraisers or when patients suffering, from a neuromuscular neurodegenerative disease visit or lab, and they hear, my boss or my lab mates talk about the research that we do, I can see the hope and excitement in their faces that there are people like my lab mates, like myself, that are, trying to work on these problems that affect their lives so profoundly. And so that has really added a component of urgency and purpose to the work that we do. That to me has been extremely humbling, an extremely humbling aspect of the training that I'm doing right now. And that while I'm just a very, very small gear in this big machine, maybe, just maybe, the research that we do might add, this tiny drop in the bucket that, might be conducive to helping folks with ALS and other neuromuscular diseases. So that's what gives me a little bit of hope that I might be able to help in a very small way.
I really appreciate my team, the people that I have worked, and they have helped me to develop all this early career that I am right now. And also something that really gives me hopes is all the advances that we have now in brain health research in the recent years. Also, plus there is a better understanding about biological underpinnings, factors associated with, and the new technologies that now we can use for designing and implementing our interventions. Like when I look back, I was like, okay, I was part of that clinical trial that right now is developing this kind of new medications that they are not now used in clinic in community-based settings. So it's like, okay, all of us, we are part of that. And that gives me a lot of hope.
Awesome. Okay. This is my favorite question. No, I don't have a favorite question. I just love talking about all of this. But some of my favorite answers came from the question that I am about to ask Arens and Laura. And it is, okay, when we, the general public, think of a scientific breakthrough, we think of, at least I think of, a scientist alone in their lab late at night and having a eureka moment. When what I know from talking to researchers is that there is a lot of failure along the way. So, can you both tell me, how do you respond to failure?
Yeah, so I mentioned earlier that failure is a common friend or enemy, depending on the days. So there are technical failures, and those are the failures that push you to get better and to improve your techniques and to learn new techniques. And that is actually very not only important for succeeding, but also very exciting opportunity, because then you can really expand your set skills and your abilities to do science. So that is one type of failure. And then there are questions that you had imagined or hypotheses that you had imagined, and you find out that they are wrong. Those are the most challenging in terms of morale, but also the most important in terms of your scientific endeavor and your scientific research, because this is how new discoveries are identified. So, I mean, it's very uncommon that you go there to confirm an idea that you had. It's more common that you, in the attempt to confirm some ideas, to verify ideas, you find through a very challenging path, a lot of turns and going back and forth until there's actually an explanation. And this is how science has actually advanced. So that part there is the one that is from the kind of the knowledge perspective, the most relevant, and the thing that we look forward, I have to say, when we face clear responses that can be yes or no, but they are clear. So that is really what we're looking for.
Yeah, absolutely. I feel the same. And to me, like, the definition of someone who's successful is someone who has tried many times and has failed many times. And it takes a lot of persistence and a lot of luck also to be successful. And so every time I, hit a wall or get a rejection or don't get a grant, I just tell myself I'm on the right track. I'm just gonna keep trying.
Okay. All right. So here is my last guided question, and then we'll get to the Q&A, and it is for Tiffany Arens again, because you've been kind of quiet tonight, and Giovanna. And it is, what is the one thing about your research that you want donors on this call to know? So hard.
I can start this. So I think what is relevant in my research is that what I'm trying to achieve is to treat ALS in kind of in targeting a mechanism that is completely unaddressed currently. There's a lot of attempt to try to cure the underlying cause of the disease, with the idea that if we can go very back into the mechanism of the disease, very back into the cause, the disease will not start. And those are very great attempts, and there are gene therapies that are being developed in that direction that are trying really to target that very specific question. But there is also a very important thing that we need to address, which is that even if we treat the underlying cause of the disease, like, for example, what we hope to do for genetic forms of ALS, the patients will be affected by weakness. They will be disabled by the degree of involvement in their motor neurons in their muscles. So we need to improve actually their strength. We need to improve how they function. And so regardless of those attempts that are so back into the cause, we need actually to treat the very important symptom of weakness, right? And weakness is something that we don't have treatments. Because we think that we have to treat the cause of weakness, but we don't have a treatment for weakness itself. We do physical therapy and we have some of these interventions, but we don't have something that actually improve the motor strength. So I think that is kind of the approach that I'm taking that will be helpful regardless of how our attempts to discover the cause of ALS go.
All right, Tiffany, go for it.
Yeah, thank you. I think I had trouble choosing just the one thing to communicate to the audience, but I think the most important thing, and I think maybe most unique to the disorder that I'm looking at, is that unlike some of the others, I guess I'm looking at chemotherapy-induced nerve damage. So we know exactly when the toxic insult's going to happen, when the neurodegeneration is going to happen. And this gives us a really excellent model to understand the mechanisms of nerve function, of nerve damage. And this really gives us a good starting point to expand the areas and the information that we learn to other neurological disorders to to help treat and to help understand the mechanisms. And that's one of the really great things about the area that I'm doing. I work particularly in neurophysiology, and by understanding exactly when the toxic insult's going to happen, how the nerves are reacting to it, and what the downstream effects is, it really gives us a good model of understanding just the neurophysiology of the humans a lot better. And that's something that I think is really exciting about my area of research.
I agree with Tiffany. Saying one thing is really hard about these topics that we are really passionate about it. But I will say that a better understanding of the relationship between mental health, lifestyles, and brain health, it will allow us to identify factors that may influence the brain health of the individuals and also the engagement in healthy behaviors. Because we can say someone is like, you need to have a healthy lifestyle, but if the person feels depressed, maybe doesn't have the motivation to do that, or maybe will be more likely to have an unhealthy behaviors. So I think that during this research and the finding for this research, it will help us also to incorporate interventions for individuals with depression and anxiety that sometimes they don't exist.
Okay. As I've said, I could talk to these researchers all night, but in the interest of answering a couple of the questions from our Q&A, I'm going to start with Peter. There's a question for you. And it is that your project sounds fascinating. Thank you, Nicole. And she is wondering how familiar you are with the critical brain hypothesis, first proposed by John Beggs and Dietmar Plenz in 2003. The control parameter is excitation inhibition balance, and it seems like it might fit well with your focus on looking at the brain holistically. And that's in the Q&A in case you want to read it.
Yeah, thanks, Nicole, for the question. Yes, I am somewhat familiar with it. We do deal with the EI balance a bit, and for folks who are not aware, you can think of the brain as kind of on a seesaw of excitation and inhibition, and you can either have too much excitation, which leads to seizures, or not enough inhibition, which also leads to seizures. So one of the interesting things in the research that I've been doing is I found that stimulation, although you think stimulation can kind of stop seizures, it turns out that sometimes they can actually make seizures worse. And kind of some of the research I'm looking at right now is kind of looking at some of that EI balance to understand, why does stimulation sometimes help seizures, sometimes hurt seizures, sometimes none of the above. So yeah, it's a very active area of research, not just for me, but for the community.
Great, thank you. We had another question about what research is being done on Parkinson's and Lewy body dementia. So I'm going to pass Parkinson's. It's sort of a broad question, but I'm going to pass it off to you, Laura, and see if you can talk a little bit about the field broadly. And I'm also going to mention that the American Brain Foundation had an active RFP. We are currently reviewing grants. We are releasing two $1 million awards in Lewy body dementia and biomarkers. And our goal is, I am not a scientist, so I hope I'm saying this correctly. Liam can jump in if I'm misspeaking. Our goal is to be able to definitively diagnose LBD in someone's lifetime by being able to differentiate it from Alzheimer's. So, okay. So Laura, can you tell us a little bit more about Parkinson's?
Well, I could say a lot, but I think a lot of these conditions we call Parkinson's disease, Parkinson's with Lewy body dementia. A lot of them we see similar, proteins and patterns of neurodegeneration in the brain. So in Parkinson's and Lewy bodies, this protein called alpha-synuclein. But everyone looks a little bit different. Some people have more dementia, and that's when we call it like Lewy body dementia. Some people have more tremor. Some people have more dystonia, like what I'm studying. And so I think understanding, why people present differently, what areas are the brain and what networks, like I'm looking into, are involved, can help us treat it better. So it's kind of all connected, and I could talk a lot about what's going on in Lewy body, but I think that's kind of one way to think about it.
Okay, great. Okay, we are right at 8. We had one more question, and I'm just going to take the time. I think this one was pointed towards Peter too. It was, what is the relationship to Tourette's if our
Um, I would say that my research is not exactly related to Tourette's. I would say one interesting thing with neurostimulation is that now there are a lot of people doing neurostimulation for psychiatric causes. So people have been implanting deep brain stimulation to help with Tourette's. Some of the research I do kind of intersects a little bit, but it's not my main focus. But there are a lot of folks, I think, here at MGH, at Baylor, at UCSF, who are doing some of that work.
Okay, so with that, thank you to all of our researchers for taking the time to talk to us tonight. You all, I don't, it's just been such a delight. I know I keep saying that to you all, but it's really been wonderful to get the chance to talk to you. Again, I want to thank our generous program partners for helping us to fund these grants. It truly would not be possible without you. And as we wrap up, I would just like to say one more time that out of every 4 applications with scientific merit that could drastically change our understanding of brain disease, we are only able to fund 1. So please consider making a donation to the American Brain Foundation today. Follow us on social media to find out more about what these researchers and others are doing. And thank you so much for your time.